Hello, good morning, class. Hope everybody is doing well. This week we're going to be doing lecture 2, which is on macromolecule. So just like last week, we will be filling in the study guide, and we'll be doing some polls along the way. Feel free to ask questions anytime you have any. So first question here. Sorry, my pen's not working. Give me a moment to fix that. Okay, there we go. Okay, macromolecule. What is the meaning of macromolecules? Well, basically, it just means that they are like really big molecules. Okay, so if you think about like H2O, right, which is water molecule, there are 3 atoms in it, 2 hydrogens and 1 oxygen. But when we are talking about macromolecules, we are talking about thousands and thousands of atoms sometimes. So they're really big molecules. That's basically what it means. So first question here, some basic chemistry. List 5 major elements that are found in living things. So if you know what they are, you can just type some out in the chat box, please. 5 major elements. Okay. Most living things are made up of these 5 key elements. So what are they? Yeah, we have hydrogen. Very good. Symbol for hydrogen is H. Carbon is another one. Yeah, there's also oxygen. Very good. Nitrogen. There's one more. What are we missing here? There's one more. Oxygen is not trace though. Oxygen is the major ones, okay? Let's see. No one has mentioned this one yet. It is phosphorus. Okay, P. So these are the major elements that are found in all living things, meaning that you would find these elements in high percentage within a living organism. Okay, so on the other hand, we have, in addition to all these major elements, we also have what we call trace elements. Okay, so what are trace elements? These are basically, these are basically elements that are needed in small quantity. Okay, so normally if you have too much, or if you don't have enough, it will result in some kind of disease condition. Okay, so trace, you just need very small amount for your body to function normally. So someone mentioned zinc and iodine. That's correct. So we can put those down here. Zinc, iodine. Right. Iodine, for example, is needed to make your thyroid hormone, right? Your thyroid gland is up here. Right? The thyroid hormone requires iodine, and they help regulate your metabolism. Calcium is another one, very good. Okay, and magnesium. That's right. Okay, of course, there are other trace elements, right? Iron, for example, that's another one. Potassium. So all these things are trace elements. Next one. Compare ionic and molecular compound by completing the table below. Okay, so the ionic compound, yes, zinc is for immune system amongst other things. So ionic compound involves metal and non-metal. Okay, so if you think about the periodic table, there is a little staircase here, right? So the stuff that is to the left of the staircase, those are the metals, and the ones that are on the right of the ladder, the staircase, we're gonna call it non-metals. So ionic compounds involves taking a metal, combining with a non-metal, and the bond that you get in ionic compound is ionic bond. Ionic bond, which involves the transfer of electron from the metal to the non-metal. Okay, so you might learn in chemistry class that metals, they don't like to hang on to their electrons, whereas non-metals, they want more, right? So this is a perfect match. You will take the electrons from the metal, transfer it to the non-metal, and in the process, you will form an ionic bond. These compounds are typically solid at room temperature. And does anybody have an example of ionic compound that they would like to share? Any example of ionic compound that they know of, maybe? Okay, so one example is NaCl. This is sodium chloride or table salts, right? So that is an ionic compound. The sodium being the metal and the chlorine being the non-metal. Okay. Let's talk about molecular compound. So for molecular compound, what we have is we have non-metal plus other non-metals. Okay, so both on the right side of the staircase. And the type of bonds that we form in molecular compounds, we call them covalent bond. Covalent bond, which involves the sharing of electrons between the non-metals. Okay, so rather than transfer, we have sharing of electron. Stay at room temperature, we have, it could be solid, could be liquid, or it could be gas. Okay? All three states are possible when it comes to molecular compound. Does anybody have any examples for molecular compound? Natalia said NaF for this one as well. Very good. This is in toothpaste, right? That's why toothpaste is salty, because of the sodium fluoride, and the fluorine is good for your teeth, right? Very good. Back to molecular compound. Mary Jo said water. Yeah, that would be a liquid at room temperature. Patricia said ammonia. Yeah, NH3. Okay, any solid or gas at room temperature? Okay, good. Carbon dioxide, it's a gas. Ammonia is it's a liquid. Yeah. Solid. Any solid molecular compound that we know for room temperature? C6H12O6, right? This is sugar, right? Glucose. Okay, so that is solid at room temperature. All right, so there you have it. Comparison between ionic compound and molecular compound. Does anybody have any question? All right, moving on. Indicate the Natalia asks, is CH4 a molecular compound? Yes, CH4 is a molecular compound. That is methane, right? Methane gas. Very good. Okay, next one. Indicate the polarity of water molecule. What is polarity? And what does the word polarity means? Well, in essence, in very simple terms, polarity basically asks the following questions. Are electrons being distributed evenly in the molecule? Okay, that's what the question, that's what polarity is. If the answer to this question is yes, that they are distributed evenly, then it's non-polar. Okay, so yes, then it's non-polar. Okay, no, then that means it's polar. So if electrons are kind of hanging around more so in one side of the molecule than the other, then it makes the molecule polar. So here is water, and it turns out that the electrons hang out around the oxygen more than the hydrogen. So it will make this part slightly negative, and then this side will be slightly positive. Okay, the correct way of writing it is like this squiggly symbol, sigma, negative, and slightly positive. Okay, so when the oxygen and the hydrogen are sharing the electrons, okay, this here is a covalent bond, right? Covalent bond. How do I know that? Well, both oxygen and hydrogens are nonmetals, right? And nonmetals, they form covalent bonds, which involves the sharing of electrons. So even though they are sharing the electrons, the oxygen actually has a higher attraction for the electron. So it's kind of hogging the electron, pulling it towards itself, therefore making it a little bit more negative than the hydrogen. Okay, it's like my daughters, right? I brought them a gift and I say, you guys got to share this gift. But then one of them, it's always hogging it, always hogging the toy and get to play with it more. So in name, they're sharing, but one actually spends more time with the electrons than the other. So that makes the oxygen more negative than the hydrogen. Okay, so overall, the water molecule is it's polar. Okay, now just because it's polar does not mean it is charged. Okay, so overall, even though H2O is polar, it is still neutral. Okay, because it doesn't have any excess electron or missing any electrons. So overall, it doesn't carry a charge. Overall, it is still neutral. Okay, it's just that part of it is slightly negative, part of it is slightly positive. Okay. Now, what is the implication of that? Well, if I draw two water molecules next to each other, okay, so let's draw a water molecule. Okay, it's like drawing Mickey Mouse. You have the ears right here. So there is your oxygen, there is your hydrogen. Okay, and then I'm going to put another one next to it, very close by. Because the hydrogen is slightly positive and the oxygen is slightly negative, there is going to be some attraction between them. Okay, I can draw another one here. Okay, again, the hydrogen slightly positive, oxygen slightly negative. There's going to be some attraction there. Okay, so these interactions between the molecules, we call that hydrogen bond in this case. Hydrogen bonds. Okay, so that gives water molecules various properties. For example, high boiling point, right? It gives it high surface tension, right? So on and so forth. And that's basically what this question is asking. Questions, anybody? How is H2O polar? Arthur asks. Okay, so again, polar means one part of the molecule is more negative than the other part. Okay. And if that happens, then by definition, it is considered as polar. Now, are you asking me how you would know if it is polar or not if I didn't tell you? Or are you asking me like what polar means? Could you clarify your question, please? How would I know? Yeah, okay. You would know. Well, okay, I would never give you a molecule and ask you whether or not it's polar or not. You will learn that in chemistry, okay? But just for those of you who are interested, when you look up the elements oxygen and hydrogen on the periodic table, there is something called electronegativity, okay? So electronegativity is basically how much does the atom likes electron, okay? To put it in simple English term. If it has high electronegativity, then it really, really likes electron. If it has low electronegativity, then it doesn't really like electrons. So if you look up the electronegativity of oxygen, it's much higher than the electronegativity of hydrogen. So when you put them together, it turns out the electrons will get pulled towards the oxygen because of its high electronegativity. So that's one way you would know. Okay, but again, I would never ask you this bio course. I just teach you a little bit of chemistry to help with what we're learning. All right. There's another question from Natalia. So basically, it's a polar bond if there is one element being distributed to other elements. I see what you're saying. Not entirely true, but we're not going to go into that. I'll wait for your chemistry teacher to clarify that for you. Whether to keep it brief, whether or not the molecule is polar or not is not entirely dependent on whether the bond is polar or not. Okay. For our purpose, what you need to know is this: water molecule is polar, but still it is neutral. Okay. And why is it polar? In our case, in the case of water, it is polar because oxygen has higher affinity, higher attraction for electron than hydrogen. Okay. Oh, that's okay. Yeah, no, don't worry about it. Okay. C. Use a diagram to explain how ionic compounds such as NaCl are soluble in water. Well, we all know if you put salt in water, like when you cook your pasta or something, the salt is going to dissolve, right? So how would it look like in a molecular level, right? Now, we know sodium chloride is an ionic compound, and the sodium is the metal part, whereas the chlorine is the non-metal part. So when you put it in water, what happens is the sodium is going to be the one that's carrying the positive charge, whereas the chlorine is the one that is going to be carrying the negative charge. Okay, so what happens is the water molecule will orient themselves such that the oxygen, right? The oxygen, if you think about again the Mickey Mouse picture here, the face is where the oxygen is, right? The ears are where the hydrogens are. So these oxygen, they're all slightly negative, and they will orient themselves to stabilize the sodium. Okay. On the other hand, for the chlorine, the water molecules would orient themselves such that the hydrogens are going to be in very close proximity to the chlorine. Okay. So this interaction allows the sodium chloride to be soluble in water. Okay, so that makes sodium chloride, right, water soluble. Okay, we call this hydrophilic. Okay, it's able to dissolve in water. It's water loving. What's the opposite of hydrophilic? That's right. The opposite of hydrophilic is hydrophobic. Okay, so if things cannot separate into positive and negative, then it won't be able to dissolve in water. Then we call it hydrophobic. So something like this, right? Something that has a lot of hydrocarbons in it. Okay, something like that. Right? This is propane, and that is hydrophobic. Okay, when you put it in water, it's not going to separate into positive and negative, and the water is not going to be able to surround it like it could for the sodium chloride, and that will be insoluble in water. Okay, moving on, we have another chart here that is comparing acid and base. So definition for acid, I'm just going to keep it very simple. Acid is something that will produce H plus when dissolved in water. Okay. Whereas base, it is something that will produce OH when dissolved in water. Okay. So OH is you probably learned this in chemistry already, hydroxide. Okay, and this one will produce H plus when dissolved in water. Now, pH is a scale that measures concentration of H plus. Okay, so if you have a lot of H plus, then it will have a low pH. And so for acid, the pH is less than 7. Okay, high concentration of H plus. Whereas for base, it is going to be having a pH more than 7, and it has low concentration of H plus. And if it's right in the middle, right at 7, then it's going to be neutral. Water, pure water is neutral. Okay, properties for acid. It is sour, corrosive, okay, conducts electricity. For base, it is bitter if you taste it. Okay, slippery if you touch it without gloves. And it also conducts electricity. Okay. Can I get some examples of acids, please? Could be ones that you are familiar with at home. Yeah, lemon juice, right? I don't know if the whole lemon is acidic. Lemon juice, acidic. Yeah, soda, right? There is the phosphoric acid in it, carbonic acid in it. Orange juice, good. Someone said HCl, which is in your stomach. Very good. Vinegar, right? All these are acidic stuff. What about base? Milk is actually basic. Okay, someone said milk earlier, I think. Water is not basic. Water is neutral. Yeah, water is right at 7. Pure water, anyways. Not your tap water. Tap water is a little bit acidic. Baking soda, yes. Baking soda. Right? Remember that in elementary school, maybe you mix baking soda and vinegar, and it creates the volcano effect. Ammonia, yeah. Okay. Bleach. All these are basic. Okay. Very good. Chlorine, not so much. Chlorine is more like an element, right? But I hear what you're saying. You hear, there is chlorine in the pool, right? Um, but it's actually talking about the bleach. Okay, the bleach contains a chlorine atom, but it's not entirely chlorine. Hope that makes sense. Yep, soap as well. Borax, yes, which is a brand of bleach, right? Very good. Human blood. Yeah, human blood is slightly basic, 7.45. Yeah, very good. 3.5 rather. Okay, so the list goes on and on, right? Excellent. Moving on. Finally, some biology now. List 4 types of macromolecule found in the body. Right? So if you have watched the lectures, you would know it's carbohydrate. Okay, proteins, yeah, very good. Lipids. And the last one is nucleic acid. Nucleic acid. Okay. The carbohydrates, the proteins, and the nucleic acids, they are polymers. Polymers of repeating units. Okay, they are like the train, right? Many, many carts linked together. Lipid, on the other hand, it's going to be not consist of repeating units, but rather it has various components to it. Arthur asked, what's a macromolecule? At the beginning, we said macromolecules are really big molecules. Okay, literally that's what it means, right? Okay, very big molecules. So in the body, there are 4 key, very big molecules, and they are here. All right. Okay. Also, right, I'm assuming you guys have watched the lecture recordings before coming to this tutorial, right? So I'm not going to be able to go into all the details that you have to know. So please watch the recordings if you haven't already. Label the diagram below with the appropriate terms to illustrate the relationship between monomers and polymers. Okay, so monomers are basically the individual building blocks shown here by these green ovals. Monomers, okay, they're building blocks. And we can link them together through a reaction. Okay, the name of the reaction is called dehydration. Dehydration synthesis. Okay, or I think I also call it condensation reaction. They mean the same thing. One of the frustrating thing about biology is they usually have several big terms referring to the same thing sometimes. Okay, so why is it called condensation reaction or dehydration synthesis? Because in order to add the monomers together, you have to remove a water molecule. So every time you form this bond, you are going to remove a water molecule. So here's 1, 2, 3, 4, 5, 6, 7 bonds. That means you would have removed 7 water molecules. Okay, and this whole chain is a polymer. Polymer. Okay, what connects the polymers together would be these bonds, right? And they are covalent bond. Covalent bond. Okay, involves sharing of electrons. We can reverse the process. We can take a polymer. So like you eat some pasta or rice, then you have carbohydrates in your system and you want to break it down to digest them back into monomers. And what is the name of that reaction? What's the opposite of dehydration synthesis? Anybody? Yeah, it involves hydration and you're going to break the bond. So it's called hydrolysis. Very good. Okay, so that is the addition of water, H2O, to break a bond. All right. Okay. Moving on. Another chart. I love charts. Complete the chart about carbohydrates. So what kind of elements do we have in carbohydrates? Well, for carbohydrates, remember we have 5 key elements, right? Carbon, hydrogen, oxygen, nitrogen, and phosphorus, right? So of the 5, what are the ones that are found in carbon? Carbohydrates. We have carbon, we have hydrogen, and oxygen. Okay. The building blocks for carbohydrates include glucose, fructose, and something called galactose. Okay, so these here, they are isomers of each other. Isomers of each other. What is the meaning of isomers? Okay, so meaning same number of meaning they are made up of same type and same number of atoms. Okay, so in this case, they are all C6H12O6. Glucose, fructose, and galactose, they all have 6 carbon, 6 hydrogen, and 6 oxygen, but they are arranged differently. Okay, so they are called isomers of each other. Okay. Now these building blocks, okay, they are all building blocks for carbohydrates, and carbohydrates are basically we call them sugars, right? So these are single building blocks, single monomers for the carbohydrates. We call them monosaccharide, right? Single sugar. All right. What do we use carbohydrates for in the body? What's the biological function of carbohydrates? Fats is another macromolecules. Okay, we'll talk about it later. Cellular energy, yep. So main source of energy. Main source of energy. Okay, think about what you just had for breakfast this morning. Maybe you didn't, but if you did, okay, whatever you ate is likely going to have some carbs in it, right? In fact, I bet it's guaranteed to have some carbs in it, right? Okay, whatever you had for breakfast, and that is going to be the major source of energy. Okay, what's another function besides being a source of energy? Many of you said cellular respiration. Yeah, we use the process of cellular respiration to produce energy, right? By burning carbohydrates. Other than that, we can store the energy, right? Storage form of energy. If you eat more than you need, you will store the extra in the liver, for example. And one last one, not so much for humans, not for animals, but for some organisms like, like plants, for example, right? Crabs and that kind of stuff. They can form, that's right, as Maphis says, formation of structural components. Very good. Okay, so structural support or components. Yeah, in plants, for example, cellulose. Good, good, good. Okay, next. Now, there will be no drawing on the test because it's not multiple choice, but it's good to, know these things. It will help you answer some of the question. So here we have 2 glucose molecules. If you count the C, there will be 6 of them. For example, like 1, 2, 3, 4, 5, 6, right? So C6, 12 hydrogen, and 6 oxygen. If you take one glucose and put it next to another glucose, what happens is you can take an OH from one of the glucose, and then you can take the H from another glucose here, and you can remove it. Right? And after you remove it, you will be able to form a new bond between this carbon and this oxygen right here. Okay, so this is going to be the new bond, new covalent bond. Okay, and as a result, what you're going to get is, okay, I'm just going to draw the hexagon and nothing else, so it's not going to get too confusing. So that's the first glucose, and then you're going to have a second glucose right here. And after forming the bond, right, you will have the new bond right here, new bond. Okay, so that's the new covalent bond that I was talking about. And in the process, you will remove an H2O. Well, where does the H2O come from? Well, we took the H away and we take the OH here, and that gives me the H2O. Okay, so this is glucose because the question told us it is glucose. So if you take a glucose and you link them together, what do we call this guy? Glucose plus glucose. What is the answer? What's the product? That is maltose. That's right. So if you take a look at the back of a candy bar, you will almost always find maltose. It's two glucose linked together. Okay, and of course there's the water molecule. Any question about what just happened here? Okay, so I can ask you on the test quiz, which by the way is next week, something like this. Let me launch the poll question. Go ahead. Okay, anyone else? Okay, just a couple more seconds in case anybody want to log in their answers. Choose something, okay? Even if you're not sure, you can choose E to let me know that you're not sure, that you're actually there. Okay, participation is important. All right. I think that's as many answers as I can squeeze out of you guys. So let's take a look at this question again. We have two monomers. Okay, you don't have to know what the hexagon and pentagon are. They basically means that these are carbons. Okay, but you know, even if you don't know that, you can still do this question. Okay, so here we have OH and OH, right? In the example we just did before the poll, we're gonna remove an OH and remove an H, right? And as a result, we'll be able to connect the hexagon with the pentagon, okay, with an oxygen in the middle. And so the first one, right, it's missing an oxygen, so that's not the answer. Right? This one it has the oxygen and there's the water, so that's the right answer. Okay, you're not going to get pentagon, pentagon, hexagon, hexagon, right? It has to be this thing connected to this. So most of you got it right. Okay, and for those of you who did not get it right, that's okay. Hopefully you understand why it is the case. Okay. Yeah, for some of you, I'm not sure why the poll doesn't show up. It happens sometimes. Thanks for letting me know. Okay, let's go back to here. Why do you remove the OH and the H? Because remember over here, right? In order to connect the monomers together, we need to do dehydration synthesis, right? And the meaning of dehydration synthesis is you dehydrate a molecule, you remove a water molecule from it, okay? So to get that water molecule, you need the OH and you also need the H. Okay, hope that answers your question, Arthur. Okay, let's see. Complete the reactions. So we have 3 reactions here. We want to get maltose, sucrose, and lactose. So the maltose is glucose plus glucose. Okay. For sucrose, right, it is also glucose, but this time we're gonna add fructose. Okay. All this is in the PowerPoint, right? And then finally, to get lactose, which is what you'll find in milk and dairy products, you would get glucose and galactose. Okay. So remember, these things over here, these are your monosaccharides. Okay. Whereas over here, these are 2 sugar linked together, so we call them disaccharide. Okay. There we go. Are we together? Anybody need any clarification on anything? All right. So next, if we keep on doing this, if we keep on connecting multiple monosaccharides together and form a really big chain, a polymer, we call them polysaccharides now. Okay, so there are some key polysaccharides that we learned in class: starch, glycogen, cellulose, and chitin. So let's briefly talk about each one of them. First one, starch. Where can you find starch? Okay, in plants, right? Okay. Example, root. Okay. So potatoes, right? Yam. All those things are very rich in starch. Main biological functions for starch is not for humans to eat it. For the plant, the reason they have starch is that the molecule is considered as a storage form of energy in plants. Okay, so like if it's in the winter, it's not a lot of sunlight, they still need energy, right? So they can tap into their starch reserve and keep the plant alive. Next one is glycogen. Okay, so where can we find glycogen? Does anyone remember where we can find glycogen? Yep, in animals, in the liver, right? Liver and muscle. Okay, and the function is that glycogen is storage form of energy in animals. If you did not eat breakfast this morning, your body is likely breaking down some of the glycogen into glucose to keep you running. All right, so we have hormones that regulate that, right? You might have heard of insulin. The opposite of insulin is glucagon, and they help regulate the glucose level in your blood. Okay, cellulose, right? Cellulose, we will also find it in plants. Okay, and this would be, you know, their fibers, like cell wall. Right, so cell would be the individual parts that makes up a plant, and then outside of the cell, you're gonna have the cell wall, like that. Okay, so the function is to provide support, right? Reinforce shape. Okay. And we also have chitins. Okay. Chitins, you will find them in exoskeleton of insects, for example, and crustacean. Okay. Crustaceans are like, you know, lobsters, crabs, shrimp, kind of stuff that has a hard shell on outside. And they again provide protection and support. All right. That's it for carbohydrate. Let's run a poll for a carbohydrate question. Select all the false statements. And so I told you some questions will have more than one correct answer, right? And it will tell you, like when you take the quiz, the question will specifically say select all the correct responses. So you will know which one has more than one answers. And this would be an example of that. So please go ahead, give that a try. Okay, just a couple more seconds. Okay. All right, let's see. First one, polysaccharides are important both for energy storage and to provide structure to cells. That is correct, right? Energy storage, we're talking about the starch, we're talking about the glycogen, and to provide structural support, we're talking about the cellulose. So that's correct. We're looking for the false statement. Next one. Glycogen is important for both energy storage and to provide structure for cell. That's not true. Okay, it does not provide structure for cells. That's the function of cellulose. Cellulose is only found in animals. That is also not true. Okay, it's found in plants. Okay, animals don't have cellulose. Glucose, fructose, and galactose are examples of monosaccharides. We wrote that down before. That is correct. Starch is a storage polysaccharide made by plant cells. That's also correct. So the incorrect statement, the false ones, would be B and C. Okay, another one. Okay, good. Again, most of you got it right. It's basically what we uh what we wrote down earlier. Okay, moving on. What do we have next? Next up, we have the lipids. So lipids is a little bit different compared to the other three macromolecules. Okay, so unlike the other three macromolecules, lipids do not have repeating monomers. Okay, instead they have components. Okay, nevertheless, they are still pretty big, so we call them macromolecules. Okay, so different types of lipids. Here's the first one. Do you recognize what this might be? This molecule here, what kind of lipid is this? What is it called? Okay, it's possible I show you a picture and ask you what you're looking at, right? Yeah, so this is fat, right? We call that triglyceride, okay, or fat. And some people even go, some of you even go a step further and tell me this is saturated fat, so even better. Right? So what is a fat molecule made up of? Well, there are two components here. Right? We have number one, the glycerol. All right. And we have the three fatty acids here. Okay. What is the main function of fat? Well, fat is also very important as a storage form of energy. Okay, so this is more for like long-term storage. The glycogen that we talked about, it's for short-term storage. This one, it's for long-term. Next one, fat is also good for insulation and protection of internal organs. And you're gonna have a little bit of fat around your heart, around your liver. So, you know, when it's minus 40 outside, the organs are gonna be nice and warm. If you fall down, you know, the fat is gonna act as a bit of cushioning, right? Just kind of like bubble wraps, right? That would protect the internal organs. Okay? Too much is not good, might not be too healthy, but it's normal to have some. In fact, it's essential to have some around the organs. Okay, so there you have it. That's a triglyceride. Over here, we have a second one. This one is a phospholipid. Phospholipid. Phospholipid, we have the glycerol again. Okay, we have some fatty acids. Okay, 2 fatty acids. And in addition, for phospholipid, as the name implies, we are going to have this extra phosphate group here. Okay. So the main function of phospholipid is that they are major. They are a major component of cell membrane. Okay. So we have a whole lesson on cell membrane later. So again, this is your cell. If you zoom in onto the membrane, you will see all these, phospholipids like that. Okay, so that makes up the cell membrane. Okay, now it's good to know, and we'll talk about this again later. It's good to know that this molecule is special because one part of it, the fatty acid part, is hydrophobic. Okay, they do not interact well with water. And then the head here is hydrophilic. Okay, so the molecule has 2 opposite properties on opposite ends. All right, next one. What is this guy? What's this thing over here? Anybody recognize that? Yes, that is steroid. Okay, and a key telltale sign of steroid is that it has 4 rings to it. Okay, so 1 ring, 2 rings, 3 rings, 4 rings. They're all fused together. And we have the components. Let me just try to draw them out for you. So we have these rings. Okay, so 4 fused rings. And in addition, we also have these things that you stick on it, okay? So those are called functional groups. Functional groups. Okay? So what do we need steroids for? Well, our body naturally makes different types of steroids. For example, hormones are steroids. Some hormones are steroids. Some hormones are proteins. So examples of steroid hormone would be the male and female hormones. What's the name of the main male hormones? Anybody know what that is? Yep, that is the testosterone. And the female version of that would be, there are actually 2, right? There's the estrogen. Yeah, and some people spell it with an O in front. Very good. And then the other one is, what's the other one? That starts with a P. Progesterone.
Progesterone.
That's right. Thank you. Okay, so if a hormone ends with O-N-E, then you will know it's a steroid hormone. Okay, there are many other examples of steroid hormones in the body, but we will learn that next semester. Okay, like aldosterone, that's another one, right? Cortisone, you might have heard of that, right? Okay. What are the functional groups again? Yeah, so functional groups are just different things you can attach to the rings that gives it special properties. Okay, there are many different types of functional groups that you can put it on, but we're not going to go into the details. Okay, so again, by putting functional groups on the rings, it gives them different properties. And another function of steroid is that they are found in the cell membrane. Okay, in the form of cholesterol. Okay, so you might hear people say, oh, cholesterols are bad for you, but that's not entirely true statements, right? We actually need cholesterol. Okay? The hormones, the steroid hormones are made from cholesterol. Okay, so we do need some cholesterol. So athlete take steroids have high testosterone. A lot of the performance enhancing drugs are steroids. Some of them are like testosterone analogs, which means they're not exactly testosterone, but they have similar chemical structure as testosterone, and in some cases that enhances their performances. Okay, but we use steroids for other medical reasons, like legitimate medical reasons. Does anybody know what are some therapeutic use of steroids might be? What kind of medical use involves steroids? Does anybody know? Severe allergies, yeah, could be. Inflammation, right? Steroid can suppress your immune system and therefore lower the inflammation. If you have a little bit of acne outbreak or something, you can put some steroid cream on, right? That's cortisone. That causes the inflammation to go away, right? So that's one example. Premature birth. Yep. I mean, if the baby comes out too early and you actually have a little bit of and we'll learn this next semester. You actually have a little bit of a lipid that your lungs make that helps you breathe. Premature babies, they haven't made that yet, right? So they have to inject a little bit of steroid up their nose to help open up the lungs, right? Okay. For pain as well, because again, the pain could be associated with inflammation, and the steroid could subside that. Yep, lots of good answers there. Very, very good. Okay, moving on. Actually, you know what? It has been an hour. I'm sure you guys are a little bit tired. Maybe we can take a quick break. Yeah, just not too long, a couple of minutes. We'll come back at 9:37, 5 minutes. Okay, and then we'll continue with this. All right. Thank you. See you in a bit. Okay. Well, since we are all kind of here, let's talk about the quiz. Okay. You will have a quiz next Tuesday, and I will post this online afterwards. Okay. It is going to be on May 24th. And like I said last time, it will be available from 6 a.m. to 11:59 p.m. Once you start the quiz, you must sit down and finish it. Okay. Even when you close the browser, the timer will continue to go down. So you have to keep going. Okay. And if your computer freezes or whatever and you restart, you will still be able to log back in and do it. Okay. It will cover lecture 1 as well as macromolecules, which is what we're doing today. And this was like last week's lecture. When I post this announcement on eCentennial, this would be a link. So on the day of the quiz, you just click on the link and it will take you directly to the quiz. Okay. There will be 20 multiple choice questions, 5 multi-select questions. No application on this quiz yet. It's the first quiz, so I'll try to keep it simple for you. And this is going to be 10% of your final grade, marked out of 30. Okay. You will not get your marks right away. Usually the marks are released on the following day. And one thing I do want to mention is that because this is an online course, you will not be able to see your quiz afterwards. Okay, so you won't be able to see which question you've got wrong, for example. Now I know that's not ideal for the situation, and many people does not like that too much, but we have to protect the integrity of the test banks, right? So for that reason, we won't be able to share with you which question you got wrong. But I will choose some questions that are got wrong by many people, and then we will take it up as a class. Okay, so that way you will still be able to see a few questions, but just not specifically your own test. Right? If you register with CALS, which is our Centers for Accessibility and Learning, and you require accommodation, let's say you have like a IEP in high school, and you need extra time for your test or something like that, then you have to get registered with the CALS and send me the appropriate documents so I can give you the extra time for all your assessment. Okay, so please contact me if you need more information on that. Does anyone have any question about quiz 1? Is this ungraded quiz that is on assessment session? No, 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 no. So the self quiz is, I think that's what you're talking about, right, Emma? The self quiz. That one you can try to just to do it. Okay, that one is not graded, but this is the actual quiz that I'm talking about, and it's happening next Tuesday. It's worth 10% of your final grade. There's going to be only one attempt for this one. Does that make sense? You won't have access to this yet. Okay, it's only going to be available, you know, on Tuesday. We usually don't have class on Tuesdays, right, guys? Tuesday is your asynchronous. So in addition to watching the lecture 3 recording next week, you also have to do quiz number 1. All right. Okay. If you thought of anything else, just pop the questions in the chat or email me. Either way is fine. Moving on, we have another table. Okay, compare saturated and unsaturated fat. Okay, so saturated fat, what do we have? For saturated fat, we have the glycerol. Glycerol plus 3 fatty acids, right? Okay. And for the unsaturated fat, it is actually the same thing. Also glycerol plus 3 fatty acids. Okay. What makes them different is the type of fatty acids that is found. So for saturated fat, we have saturated fatty acids. And let me zoom in a little bit here and try to fit it all into the box. Okay, there we go. So we have saturated fatty acids. Okay. And what that means is the carbon atoms are bound to a maximum number of hydrogen atoms. Okay. They are saturated with hydrogen atoms. And you will have no double bond. No double bond in the fatty acid. Okay. On the other hand, for the unsaturated fat, we have the unsaturated fatty acids. Okay. Carbons, in this case, the carbon atoms are not bound to a maximum number of hydrogen atoms. Okay. And some double bonds exist. Could be 1, could be 2, could be couple. In the fatty acids. Okay. Give you a moment to copy what you need. Next row is the state at room temperature. What do you guys think? Saturated fat, are they solid or liquid at room temperature? Solid or liquid? Yeah, they're solid at room temperature. Right. Again, there are exceptions, right? Not all saturated fat are going to be solid, but in general, they're going to be solid. Okay, and it's because there's no double bond in the saturated fatty acids. So, you know, it's all straight. and they can all pack together really closely. Okay, so when they pack together really closely, then we get the solid state. Whereas for the unsaturated fat, they have the double bond, and double bond creates these kinks, right, in the structure. Okay, so they're not going to be able to pack really closely together because the kinks kind of get in the way. And so they are mostly going to be liquid at room temperature. Okay. Help me out here. Give me some examples of, natural sources of saturated fat. Where do we find them? Anyone? Okay, they're generally derived from animal fat. So give me some examples. You must know some examples of saturated fat. You might even have some in your fridge. Yes, butter. Butter is saturated. What else? What kind of oil though? There are so many different kinds of oil, right? Peanut oil, canola oil, flaxseed oil. What kind of oil? Yes, coconut oil. Lard is another one. Lard. Can use lard to make your pastry, makes them fluffy and very tasty, but probably not too healthy. And coconut oil, right? So coconut oil, if you ever seen it, it's kind of like this white paste. Okay, even though coconut is not an animal source of fat, it is a type of saturated fat, right? So there are some exceptions, right? Okay, for the other guy, unsaturated fat, these are generally derived from plants, right? Okay, so examples. Give me some examples of this. Yep, olive oil, my avocado oil. Okay, so these are again derived from plants. Okay, very good. So which one is more healthy for you? Okay, if you have to pick one to eat, put on your toast, which one is better?
Unsaturated.
That's right. This is the healthier version, right? Okay, but I can't really spread olive oil or avocado oil on my toast. It's just make it soggy and drench all over the place. So what people do is they actually artificially convert the unsaturated fat into saturated fat, okay, using what's called hydrogenation. Okay, so in hydrogenation, you artificially add hydrogen to the unsaturated fat to make it into a solid state. Okay, so in the supermarket, you will see these, these margarines, right? Say, oh, it's derived from avocado, but actually it's not that healthy, right? Because even though the source might be avocado, it's a highly industrialized chemical process that cause it to become the solid state, right? And it's not as healthy as it claims. Arthur asks, unsaturated fat is healthier, but why? It's healthier because of its structure, right? Like they don't tend to clump together, right? Because of the kinks. And saturated fat, they come clumped together quite easily. So imagine having this in your bloodstream, right? In your artery, they might start building up easily and clog your artery. And that's one of the reasons why it is not as healthy. Okay, now in the process of doing this, it also generates what we call trans fat, right? And trans fat is like the worst kind of fat ever because your body cannot break them down. And they just accumulate in your system, and it causes a lot of cardiovascular problems. Fortunately, I think almost 4 or 5 years ago now, trans fat is actually banned completely in Canada. Okay, so no food can contain trans fat, not fast food, not processed food. It's completely banned, so that's a good thing. Butter is healthy. Again, right? Butter is saturated fat, so I wouldn't say it's healthy. I mean, it's okay to have some in moderation, but definitely. Oh, and again, right, just because it's natural doesn't necessarily mean it is healthy, right? The processed stuff can be unhealthy for other reasons, but just because it's natural does not necessarily mean it's good for your body. Okay, there we go. That's it for fat. All right. Any questions? Any other questions? You guys are asking some very good questions. Any other questions about fat before we move on to proteins? Proteins is the one that is like intense, right? So just want to make sure you guys are doing okay before we move on. I hope this is okay, right? I hope that the pace is not too fast and this format is suitable for you. That you do some study on your own and then we come together and take this up, right? Hopefully that gives you some flexibility in terms of when you come to class and stuff like that. All right, next part, proteins. Label the different parts of an amino acid. So amino acids, they are the monomers of proteins. Monomers just means building blocks, right? Monomers of proteins. How many different types of amino acids do we have in the body? Does anybody remember? What's the magic number? If I ask you a multiple choice question, how many amino acids do we have in the body? Yes, so 20 types of amino acids. Okay, even though there are 20 different types, they all kind of look very similar. Okay, and you should know what the parts are. I think there might be a question on that. Oh, by the way, the quiz and test for that matter, your quiz is not going to be the same as someone else's, right? Okay, we as in like me and other people who teach us the course, we've created literally thousands of questions in the test questions bank. So it's going to be randomized for each person, just so you know. Okay, so that if you do talk about it with someone else afterwards, you don't freak out that, oh, I didn't get that question, did I miss it? It's probably a different test. Okay, so here is the general structure of an amino acid. We have what we call an amino group. Okay, and on the other side over here we have another group. Okay, and this group is what we call the carboxyl group. Carboxyl group, okay, which is acting as an acid, whereas the amino group, it is acting as a base. Oops, that's the wrong purple. Okay, so that's why it's called an amino acid. It has both the basic group and the acidic group. Okay, and we talked about acid and base earlier in one of the charts, right? There is going to be a central carbon, and then there's going to be a hydrogen. And what distinguishes them, what distinguishes one amino acid from another is this R group. Okay, this is called a side chain. All right. So all of the 20 amino acids will have the amino group, central carbon, hydrogen, and the carboxyl group, but their side chain is going to be different. Okay. So we can call this like side chain number 1, side chain number 2. So if you put one amino acid next to another amino acid, and then we want to connect them, just like before, we are going to remove an OH from the carboxyl group of one amino acid. We'll remove a hydrogen from the other amino acid. So these things comes out and forms water, right? And afterwards, we are going to be able to form a new bond, a new connection between the carbon and the nitrogen, like that. Okay, and this bond is what we call the peptide bond. Okay, Now you don't have to draw this next part, okay? In fact, don't do it. I'd rather you just watch what I do and try to understand what I'm saying, okay? So I'm gonna redraw this one. And we're gonna have the NH like this, CH, and then this is R1, right? Then we're gonna have C double bond O, just like here. And then we're gonna have our peptide bond that connects to the other NH. This is R2 now. Okay, so you see, we have a peptide bond right here. This is the first amino acid. This is the second amino acid. Now keep watching. What your body can do is you can then connect this with another one. Connect it with another amino acid. I will call this R3, side chain number 3. Right, and then we can keep on doing this. Side chain number 4. Right, and then side chain, so on and so forth, right? Number 5. And you can see, I hope you can see that, the structure over here, this part here, it just keeps on repeating itself, right? Look at it. It's just the same thing over and over again, right? We have a C, CH, CO, NH. CH, CO, NH. CH, CO, NH. It just keeps on repeating. So if I keep on doing this, you will eventually form, you know, a very long polypeptide. Okay, so this whole thing is a polypeptide. Remember, before we have monosaccharides, then we get the polysaccharides, right? And over here, we have the amino acids, which when we link many of them together, we get the polypeptide. Emma asked, what is side chain? Okay, so side chain is different for each of the amino acid, right? Let me just quickly grab a picture from the internet to show you. 1 second. Image. I need to find something that's easier. Okay, this one is good. Sorry, just copying it over. All right. Oh, come on, really? There we go. Okay, so you see, these are all the different amino acids, right? And all of them has the CH and then the COOH and the NH2, right? They wrote it as NH3 and, you know, with a negative here, but it's the same thing. Right? So that's a side chain. That's a side chain. That's a side chain, right? So the side chain is what gives each of the amino acid its unique property. Okay, and that's what differs one amino acid from the other. Some side chains will make the amino acid nonpolar. Some side chain will make it charged. Some side chain will make it polar, right? And so on and so forth. Does that make sense? Okay, so here you have your polypeptide. And then, you know, the point I'm trying to make, and it's an important point for you guys to understand, is that the backbone does not change. Okay, but the side chain is what's changing, right? Okay, so I'm just gonna write here the polypeptide backbone does not change. Okay, it's the same thing. Okay, and then again, right, this is your amino acid number 1. This one here is your amino acid number 2. Amino acid number 3, so on and so forth. Okay, and you might have like hundreds and hundreds of these, or it could be as short as 4 or 5 amino acids. It really depends on what kind of protein you're trying to make. Okay. Draw the peptide backbone of multiple amino acid linked together. Oh, okay. So this it's what I did here. Okay. I just forgot that there's a question for it. So there, I'll put it here, and then and then that would be it. Okay. There we go. Okay, you can copy that after if you want to. Okay, now it just looks really, really weird. But that's okay. I hope you guys understand what I'm trying to say. So this is very, very long, right? Okay, there we go. Okay, complete the table about proteins. Okay, elements that are found in the proteins. Okay, so we have our usual ones, the carbon, the hydrogen, the oxygen. There's also nitrogen that is found in the amino side. And excuse me, and there is going to be trace of sulfur. Okay, a little bit of sulfur. If you look here, one of the amino acid, cysteine, it has sulfur in it. Okay, nothing else has. Oh, the methionine also has a sulfur here. Okay, so some of them has sulfur, but not a lot. So traces of it. All right. Building blocks. We have the 20 different amino acids, right? 20 amino acids. 12 of them are non-essential, meaning your body can make them, and 8 of them are essential, meaning you will have to consume it from your diet. Biological functions. What do we use proteins for? Some of them are hormones. Okay, so hormones can be lipid, such as testosterone and progesterone, but hormones, in fact, most of them are protein hormones. So an example of protein hormones that you might heard of is insulin. Right? It helps you, regulate blood sugar. After a meal, the insulin would help you absorb those glucose from your blood. Does anybody know another protein hormone by any chance? Right. Another one that like if we have any mothers here in the class, you might have heard of oxytocin, right? Oxytocin. Anybody know what oxytocin is?
No.
No. Okay. Oxytocin, yeah, it's for milk production, right? And it induces labor, right? Okay. In fact, if you are overdue, for example, they might give you artificial oxytocin, and it's gonna help you start the contraction. Okay. All right, that's glucagon is another one. Very good. Okay, so that's hormone. Another function of protein is that they form enzymes. Okay, enzymes, they catalyze reaction, they speed up chemical reactions. We also need them for fuels, okay, and structure. Okay, such as your hair, fingernails, okay, they all are made up of proteins. All right, moving on. Complete the table about the structural levels of proteins. First one is already done for you. So primary structure is referring to the sequence of amino acids. All right. So if you take a look at a polypeptide, you would have one amino acid, another amino acid, right, and then another one. And then you might use the same one again. It doesn't really matter. Sometimes they repeat, sometimes they don't. Afterwards, you are basically going to have a very long chain of these amino acids, right? So if you read the sequence from one end to another, that order that you just read, that would be called a primary structure of the proteins, okay? And what holds them together is the peptide bond that we drew earlier. Yeah, so the illustration for primary structure, again, is just the amino acids all linked together like this. Okay. Secondary. Okay, so these amino acids, once they link them together, some of them is going to form these helices, and then others are going to form these zigzag patterns. Okay, so secondary structure are referring to the formation of alpha helix and beta pleated sheets. Okay, and how we stabilize them is through hydrogen bonds. From the backbone. Okay. So remember what I drew earlier just a couple minutes ago? You have the peptide backbone here. Okay, so one part of the backbone might be able to form some hydrogen bonds with another part of the backbone. Okay, and these hydrogen bonds is what's stabilizing the secondary structures. Okay, we have the tertiary as well. Okay, so for the tertiary structure, we are referring to the overall 3D structures. Okay, and the overall 3D, we can call 3D shape. Okay, the overall 3D shape is also known as the conformation. Conformation. Okay, and what holds them together would be the interaction. Interaction between the side chains. Okay, so in the PowerPoint slides, there is one slide that shows you all the different types of interactions. Some are ionic, some are covalent, some are hydrophobic. Okay, you can refer to that slide and see for yourself. And in terms of an illustration, what happens is these things will fold up together, right? It's really messy drawing, but the point is they will fold up into some kind of 3D shape. Okay, and the kind of 3D shapes that you get, that's what we call the tertiary structure. Okay, now many proteins will stop at this level, but some of them will go on and form a quaternary structure. And that happens when two or more polypeptide come together. To form a complex. Okay. And what holds them together would be the interaction between each of the polypeptide. Okay, so you might have, two pink subunits, and then you might have two blue subunits. Okay, and that forms like an entire complex that has some other function. Yeah, exactly, Mavis. Like hemoglobin, right? You have the alpha chains and the beta chains, and it's like one big complex. Okay, that's that. All right, moving on for nucleic acids. Complete the table about nucleic acid. All right, so what kind of elements do we have? A little bit of everything here. Okay, we have the carbon, hydrogen, oxygen, nitrogen, phosphorus. Okay. And the building block for nucleic acid is what we call a nucleotide. Nucleotide. Okay. And what do we need it for? Well, nucleic acid contains genetic information, right? Right. It contains instruction to make proteins, for example. How do you know which amino acid to connect together, right? How do you know, how many amino acids to use for a given protein? All that information is encoded within your DNA. Excuse me. Okay, next part. Identify each of the following. So this whole thing is a nucleotide. Okay, and the nucleotide has three parts to it. there is a phosphate, a PO4, -3. You might learn that in chemistry. We have a sugar, which might be ribose or deoxyribose, depending on which type of nucleic acid you're talking about. And we have a nitrogen base. Oh my goodness. Nitrogen base. Or a nitrogenous base, sometimes it's called. All right, so together, it makes up a nucleotide. This whole thing is a nucleotide. Okay, the building blocks for nucleic acids. Okay. Identify each base as purine or pyrimidine. Okay, so the first three, they are the pyrimidines. Okay. Pyrimidine. Pyrimidine has one ring in their structure. Okay, so we have the cytosine, thymine, and uracil. These are considered as pyrimidine. And over here, we have the purines. Purines, they have two rings. Okay, and if you're like, well, how am I supposed to memorize that? Well, you can remember this this phrase, pure as gold. Okay. So purines include the adenine and the guanine. Okay, so adenine, guanine, they are purines. Do you know what condition, if someone has this particular condition, they have to avoid consuming purines? Does anybody know what that is? Oh, Yamir, thanks for letting me know. Don't think I could fix that though, because I lost the original file, but that is good to know. Thank you. Do you guys know what condition, if you have this condition, you have to avoid having purines in your diets. And you would get a lot of purines from like alcohols, for example, especially beers and red meat. Anybody know? Goat? Anybody heard of goats before? Goat is something that happens when you have buildup of uric acid, okay, in your joints. And of all places that the uric acid could build up, one of the places that they usually build up is like in your toes, in your big toe, particularly. Okay, so for people who are generally speaking, this is for people who are like a little bit older, okay, and they have this gout condition. It can be so painful that they wouldn't be able to walk properly. They may need a cane or they may need bed rest. But having beer, for example, and red meat, that increases the amount of purine in the system, and that makes the condition worse. But it's not just restricted to OH. I have a friend who had gout since he was in his late 20s. Okay, and he has to cut out all these food that has very high purine contents in them. In any case, these are the pyrimidines and these are the purines. And as you know, in terms of the base pairing rule, the purines will pair with the pyrimidines. So A pairs with T or U, whereas G pairs with C only. Okay, so let's finish the chart here. Nucleotides for DNA, we have the phosphate plus the deoxyribose. And also a nitrogen base, nitrogenous base. Whereas the RNA, that's the same thing. We got the phosphate, but this time is the ribose sugar. It has an extra oxygen. plus nitrogenous base. What kind of base can you have for DNA? It could be A, C, T, or G. Okay, 2 purines and 2 pyrimidines. Same thing for RNA, but instead of the thymine, it's going to have a uracil instead. DNA, I'm sure you've seen it on TV or in textbooks. It's a double helix, okay, and it is complementary. Okay, one side, if you know one side, you can figure out what the other side is. Whereas for the RNA, it is single-stranded. Okay. Now, we don't have time to go through the long answers. I'll post the answer online afterwards, so you can take a look at themselves. Take a look at them yourselves. But let's just go through some of these application questions. Now, I am aware of the time, and I think you guys probably have another class in the afternoon, right? So if you have to go, feel free to go. You can watch the recordings afterwards. But you know, if you want to stick around, feel free to do that as well. Okay, so first question: in a certain species of animal, 30% of the base in its DNA are adenine. Find the percentage of other bases. Okay, so because A pairs with T, the amount of A that you have will equal to the amount of T in the body, right? So if you have 30% adenine, that means you have 30% thymine as well. They have to be the same. Okay, so 30% plus 30% all together gives you 60%, right? Which means what percentage makes up C and G? If 60% makes up A and T, what percentage makes up C and G in the body? Yeah, 100% minus 60% is 40%, right? And we know the amount of C and G would be the same, which means I take this number and divide it by 2, we will have 20% C and 20% G. Okay? Do I have a, no, I don't have a question for that. All right, but hopefully that makes sense. What was C and G again? I'm sorry, could you clarify what do you mean by that question? Like, as in, what do they stand for, or what? Oh, the cytosine and guanine. Okay, so if I go up here, right, you can see, right, that's the individual symbols and what their name stands for. Okay. All right. Next question. Does changing the primary structure of a protein always have an effect on the shape of the protein? Justify your answer. Okay. So not always. The answer is not always. Okay. It depends on what changes were made. Okay. Sometimes, remember, we have 20 amino acids, right? Here are the 20 amino acids. So, you know, let's just say, for the sake of discussion, the original amino acid is glycine. Okay. And then you can see the side chain is H, right? And if for some reason there's a mutation that causes it to become adenine, okay, and you look at glycine, which has an H, and adenine has CH3, well, these two look very similar, certainly more similar than something that has like a ring in it, right? So if you are able to replace one amino acid with another amino acid that's very similar in structure, right, you might not alter the structure at all. Okay, you might not alter the shape of the protein at all. Okay, So, does changing the primary structure of a protein always change the shape? Again, not always. It depends what the change was, and also it depends on where the changes took place. Okay. Remember, your amino acid, your polypeptide rather, is really, really long, right? Okay. So if the change happens at the very end, then maybe it's not going to have as much of an effect as if the changes happens in the middle, and that middle happens to be a section that will form a helix. Okay, so not always. It depends on, number one, the type of change that you're making, right? And once again, just for illustration, if you're changing from like glycine, which is non-polar, to something that's polar, like serine, then yes, probably is going to have a very big effect. But if you're just switching to something that looks very similar, right, like serine and threonine looks pretty close together, right, maybe you're not going to have an effect. Secondly, it depends on where it happens. If it happens towards the end, right, maybe it's going to be okay. Does that make sense? All right, I hope it makes sense to the rest of you as well. Your digestive system uses many enzymes that break down polymers into your food. Breaks down polymers in your food down to monomers before absorbing them. Enzyme names often end with the suffix ase. For example, lactose are broken down by lactase. A generic name for these enzymes is hydrolases. Explain the basis of such name. Well, it's not too difficult because we know going from polymer to monomers, the process is called hydrolysis, right? Okay, so to perform hydrolysis, we would need to have hydrolase. Okay, enzymes that does hydrolysis. Essentially, that's what it means. Okay. Okay, next we have the fill in the blanks, which again, I will just post the answer for you to check. You can probably do that on your own. You can go if you want to go. I'm just going to go through a few more practice questions with all of you. And then that will be it. So stay if you could. If not, I'll see you next week. All right. What is a chain of 25 amino acids called? Do you guys want to do the polls, or you want me just to tell you the answers? Let's do a poll on whether or not you want to do a poll. Okay. Launch the poll. Okay. So if you want to do poll, say yes for 1 for A. B is no polls. Just tell me the answer. Give me the answers, and I'll do what the majority of you wants to do. Do we need to draw eyes on quiz? No drawing. Okay, no drawing on the quiz. Okay, we have an overwhelming of you asking to do the polls. All right, we will do the polls. Okay, if you don't want to do the polls, that's okay as well. You can just come back and look at the results later. Okay, let's do the polls. Relaunch the poll. Okay, so we will keep it at 1 minute per 1 minute per question. Okay, so that way we can do more. What is a chain of 25 amino acid called? Tell me what it's called. Okay, I get my usual 17 response. I think that's usually how many people respond. I'll move it on. That's correct. The answer is polypeptide. Okay, amino acid makes protein, right? So polypeptide. Not polysaccharide. Polysaccharide, it's sugar, right? Okay, and nucleotides are building blocks for DNA. Okay, next one. Relaunch. Okay, so let's take a look at this diagram here, the one that we drew earlier. There we go, the peptide bond, right? How do we form the peptide bond? We have to connect the carboxyl group here with this one. What is this called? Well, it's named here, right? Amino group. So carboxyl group and amino group. The answer is C. Okay, we do not connect the side chains. The side chains are never connected. All right, so if you see this question on the quiz, make sure you choose carboxyl and amino group. Okay, for those of you who chose B, have I convinced you by showing you this picture of why the answer is carboxyl and amino? Any question about that? Okay, another one. Go ahead. The correct answer is quaternary, okay? But because it has multiple polypeptides coming together, right? Good. Here's another one. Okay. Not as many response here. This one is a little bit tricky, right? You have to do some reading, right? Okay, so let's see. Pancreatic cell make insulin, okay? And if you don't know what insulin is, the question tells you it is a type of protein, okay? So these cells use something to synthesize insulin by the process. So protein, what do we use to make proteins? Do we use nucleotides to make protein? No, we don't. Is it fatty acid and glycerol? Nope. Is it saccharides? Nope. Not saccharides again, right? We use amino acids to make protein, right? And how do we connect the amino acids together? The same way we connect any monomers together, through dehydration synthesis. That's why the answer is E. Okay. Moving on. Again, you can get access to these questions by looking at the recordings later on, right? Just like skip to the end of the recordings and you know, you can see them. Another one. Most of you got it right. Purines have double-ringed structure. If we go back to the picture here, we can see that the purines, right? Purines have 2 rings in it, okay? One way I remember is purine has 2 syllables, 2 syllables, 2 rings, okay? And just to mention something else here, adenine's bonding to thymine is stronger than, oh, this, sorry, grammatically it doesn't make sense, stronger than guanine to cytosine. That is not true, okay? It's actually the other way around, okay? G to C is stronger than A to T. Okay, I don't remember if I mentioned why in class, but G to C has 3 bonds to it, whereas A and T only has 2 bonds. Okay, so this is the stronger one. Something to keep in mind, okay, just in case. All right, I have a couple more if you guys are up for it. Okay, this is a multi-select question. Okay, let's take a look at this one. We're looking for the false one, okay, not a feature of DNA. So nucleotides within a single strand are connected by covalent bond. That is true. That is true. Covalent bond always connects the monomers together. Adenine forms a base pair with uracil. That's not correct. There is no uracil in DNA. It's supposed to be thymine. Okay. Complementary strand, that means the left strand and the right strand, are held together by hydrogen bonds between the phosphate group. That is not true, okay? Between the nitrogenous space. Okay. Complementary strands are anti-parallel, run in opposite direction. That is correct, okay? If you look at the PowerPoint slides, one strand is always upside down compared to the other. All right, so the incorrect statements would be B and C. Okay, maybe just a couple more, and then I will let you be on your way. Okay, give me 1 second. Just have to copy them over. Okay, this one is a little bit different than the ones we have so far. And this one requires you to think a little bit, huh? Go ahead. Oh, sorry, I forgot to put the A, B, C, D. A, B, C, D. If you click the wrong one, that's okay. But like, just in your mind, right? Make sure you know which one you've picked. Okay. Okay. Anyone else? Again, the results might be a little bit off because I didn't have the A, B, C, D. Like in your mind, you might think this was A, B, C, D, right? But anyways, a chemical reaction occurs slowly at 37 degrees. When some molecule is added, the reaction speeds up significantly. Which of the following is the monomer of that molecule? So what can we add to speed up chemical reactions in the body? Anybody know what the word I'm looking for is? What can you add to speed up a chemical reaction within the body? That's right. The answer is enzyme, right? And what are enzymes? Are enzymes carbohydrates, proteins, lipids, nucleic acid? What are they? They are proteins. So what is the monomer of proteins? The answer would be nucleic acid. Okay, nucleic acid. So you see, this would be an example of like an application question, right? Where you have to think, multiple layers before you get to the answer. All right. Okay, so that is it. Remember the quiz is next week. Do take advantage of the practice quiz. But keep in mind that the practice quiz are just practice. They are not going to be as difficult as the actual quiz. So if you get perfect on the practice quiz, don't think, oh, I'm gonna get really good grade and I can stop studying. Definitely not the case. Try to have like a one-page help sheet with you when you take the quiz, right? So you don't have to fumble around to find information if you need that. But at the same time, don't use that as like a psychological safety blanket, right? Just because you have a help sheet doesn't mean you don't have to study. Okay, you still have to know things on top of your head. Otherwise, you'll run out of time. Okay, so sorry about going over time, and thank you for sticking around. If you have any questions between now and the quiz, feel free to contact me. Otherwise, I will see you on next Thursday. There's another question. Why is A the answer again? Okay, so first of all, you have to understand that the enzyme is the one that is speeding up the chemical reaction, right? Okay, the definition of enzyme is a biological catalyst. So once you establish it is the enzyme that's speeding it up, we have to know that enzyme is made up of proteins, and the monomer for protein is amino acid, and you have to know that amino acid looks like this, right? Okay. Mohammed, I see you're on mute. Sandeep, I see you're on mute. Go ahead, please.
I sent you an email about ISP.
I replied to you already.
Yeah,
yeah.
Yeah, I got it. But I have a question. Like, you will set up when you set the time.
All your assessment will have 1.5 times automatically. Yeah, I set that up.
Do I need like remember you for each and every?
You don't have to. You don't have to. Yeah, it's okay. I automatically set it up so that it does it for all the assessments.
Okay, thank you so much.
Yeah, no problem. Sandeep, I see that you also unmute. You have a question, or you just unmute by mistake? Okay, all right. All right, bye everyone. Have a fantastic day and week. See you next time.